My name is Erika Ross. I'm a third-year graduate student in the Neural Engineering Lab at the Mayo Clinic, and I'm going to talk about our work looking at Fornix deep brain stimulation and showing how it directly modulates medial limbic and corticolimbic circuit connectivity. I'd like to thank the organizers for putting this meeting together. It's been a lot of fun and very interesting to learn about some other stimulation modalities outside of deep brain stimulation. So I think a lot of you are very familiar with deep brain stimulation and that it's been shown to be effective in disorders like Parkinson's disease and essential tremor, dystonia. More recently, it's being investigated in depression as well as obsessive compulsive disorder, but also Alzheimer's disease. This was started actually very serendipitously. They were targeting the hypothalamus in a case of obesity and trying to use this target for weight loss. But during this procedure, this patient experienced intraoperative memory recall, this describing events that happened long ago in his past. And the story goes that based on these experiences, they looked at the anatomy and saw they were very close to the fornix, which is a well-known structure involved in memory. So they started this phase one trial of deep brain stimulation targeting the fornix. In this study, they showed that patients after 1 year of deep brain stimulation had increased glucose metabolism in cortical, subcortical networks that are involved in memory, specifically hippocampal structures. Several years later, they've also shown that 2 patients out of the 6 had increased hippocampal volume after 1 year. So for the first time, as far as I know, Alzheimer's disease patients have had this increase in hippocampal volume, which is we know that Alzheimer's disease patients typically have decrease in hippocampal volume. And so this is a very interesting question. Why is deep brain stimulation working in Alzheimer's disease? Why is deep brain stimulation really working in anything? There are all kinds of hypothesized mechanisms of action in various diseases based on time frame of the effectivity of it within different diseases. Things like neurotransmitter release early on, neurogenesis later on in these various diseases. But really, our question was, where does Alzheimer's disease fit into this? What kind of circuit involvement are we seeing with this? And in the neurosurgery department at the Mayo Clinic, in the Neural Engineering Lab, we have a very nice translational neurosurgery and imaging program. We're able to perform intraoperative fMRI in patients as well as in a non-human primate model. And in this talk, I'm going to talk about our swine model, where we've developed an MR-safe Lexcel targeting system, stereotactic head frame, that we're able to implant the Medtronic lead and stimulate and image, perform fMRI during stimulation to see which circuitry is involved during deep brain stimulation. And with this setup, we've seen very consistent results between species. So in Parkinson's disease patients, we see increased BOLD response in motor cortex, pontine nuclei, and cerebellum, which is very well conserved into our non-human primate model as well as our swine model. So a little background here. The medial limbic circuit, or the Papez circuit, consists of the hippocampus, which projects through the fornix to the mammillary bodies, up through the anterior thalamic nucleus, through the cingulate gyrus, down through the parahippocampal gyrus, entorhinal cortex, and back to the hippocampus. And the target for these studies is actually anterior and parallel to the fornix here, because disruption of that white matter tract has actually been shown to cause memory deficit. And so the idea is to use current spread into the fornix to activate this circuit. Based on this, we targeted very similarly to this within our pig model. So you see we've targeted anterior and parallel to the fornix. So using this model, we have looked at BOLD increases using fMRI to see which parts of the brain we see increased BOLD response following Fornix stimulation. And we've seen areas that you would very much expect, areas within the medial limbic circuit, as well as corticolimbic circuit structures. So the nucleus accumbens, prefrontal cortex, amygdala, these areas that are very well connected. We also see a very similar BOLD response in these areas, so prefrontal cortex, nucleus accumbens, hippocampus, as well as the inferior temporal gyrus. We see similar BOLD response even with increased amplitude. So we're interested in why we see these increased BOLD responses in these areas. What specific neurotransmitters were related to these increases in BOLD. And our lab has a setup where we can actually real-time monitor neurotransmitter release with stimulation. We have this system called our Wireless Instantaneous Neurochemical Sensing System, or WINCS, which is a fast-scan cyclic voltammetry tool that we use to monitor real-time neurochemical changes in the brain. And so using this system, we implanted the carbon fiber microelectrode into the nucleus accumbens specifically to see what neurochemical changes we were related to this BOLD response in the nucleus accumbens. And here we show this biphasic dopamine release. So you can tell on the 3D color plot here, the dopamine oxidation current here and reduction current here. Here I have the oxidation current plotted, where we see this biphasic dopamine release, which has been previously reported in the literature following hippocampus stimulation. So we're seeing dopamine release in the nucleus accumbens with Fornix stimulation. So this interplay between the medial limbic and the corticolimbic circuit. And we were really interested to see what global changes happened if we changed this local neurotransmitter release in the nucleus accumbens. So we came up with this setup where we performed intracerebral receptor antagonism in the nucleus accumbens, and then monitored fMRI following drug injection. So here we used two different drugs. We used a global dopamine receptor antagonist as well as a global glutamate receptor antagonist to see what changes occurred following nucleus accumbens receptor antagonism within these circuits following Fornix stimulation, to really look at the interplay between these circuits and see what changes we could see. So with dopamine antagonism as well as with glutamate receptor antagonism, we saw a significant decrease in the amygdala and hippocampus following drug injection with Fornix stimulation. And we saw approaching significance in the prefrontal cortex and the nucleus accumbens with both drugs. And with just the dopamine receptor antagonist, we saw it was approaching significance in the nucleus accumbens. This is a very small N number at this point, so these values, this is why I've presented this with this p-value 0.1, but it is approaching significance in these dopaminergic or corticolimbic circuit structures. Finally, we were interested in looking at the functional connectivity with Fornix stimulation. And so we came up with a model using prefrontal cortex, nucleus accumbens, hippocampus, and looking at the reciprocal projections between all three structures, building 7 different models, looking at these different structures. And using a Bayesian model selection, we found that the model that won was this model in particular, showing that Fornix deep brain stimulation modulates each node individually, as well as the reciprocal projections between each node. Following drug injection, we saw that the dopamine antagonist significantly increased activity between nucleus accumbens and hippocampus and decreased between hippocampus and nucleus accumbens. So in summary, Fornix stimulation resulted in predominant and selectively potentiated hemodynamic increases in medial limbic and corticolimbic structures. Direct electrical stimulation of the Fornix acts as a driving input into the medial limbic and corticolimbic circuit nodes. Fornix stimulation results in biphasic dopamine release in the nucleus accumbens. And finally, Fornix stimulation-induced hemodynamic increases in the hippocampus and amygdala are at least in part dependent on circuit involvement via the nucleus accumbens. I'd like to thank the director of the Neural Engineering Lab, Kendall Lee, as well as Paul Min, who's here today, and Will Gibson for his work with the dynamic causal modeling, who's also here today, and they both have posters, and we'll talk a little bit more about the work we're doing intraoperatively in the clinic as well. I'd also like to thank our funding sources, the NIH, as well as the Granger Foundation, Mayo Foundation, and the Sidney Luckman family for my graduate fellowship. Thank you all for your attention.